RNAct, enabling proteins for synthetic biology and bioanalytics.
Well, hello and welcome to the webinar on how to produce and store biological data to enable data-intensive research. My name is Jose and today I'm going to be teaching you some of the things I know about this. And I would like to start with this slide. Data is gold, don't waste it. Why do I say this? Because money starts, with money comes everything. Money is invested in our education, and once we have become scientists with this education, then governments and other funding organizations put money into us again so that we can do our science. From the research that we do, we generate some data, and traditionally from this data you only care that it gives you a publication, but you don't care that much about the data itself. It was just a mean to get this publication. But this data here is still very, very useful and should be used, stored and used for different purposes. And with this we come to biological databases where this data can be stored. And we have a few examples here of databases that are relevant for, contain different kinds of information, as you see here. And it is very useful because as a researcher that maybe doesn't know how to program, but it still needs information, you can look into, for instance, UniProt and get for this one specific sequence that you need certain information. We're actually going to go to UniProt afterwards and we'll expand into which kind of information you can find. But very interesting, if you're a computational scientist and you might be interested in getting this kind of data from multiple databases at a level that is not sequence by sequence or entry by entry. And this might be done because you're doing some data analysis or some analysis, some review of what has been done for, you name it, any kind of type of data. But also, and this is gaining a lot of importance in the last few years, it is being used a lot for artificial intelligence. Very often now for diagnosis, if someone is suspected to have cancer, it's not going to be the physician looking at this mutation here is this and that. It's usually an algorithm, and this is improving our life a lot. But very recently, there was a great advance in artificial intelligence for biology, which was the creation of AlphaFold. So in November 2020, we woke up with the results of the 14th critical assessment of protein structure predictions, or CASP. And in this assessment, we saw how there was basically the problem on how, if you have an amino acid sequence, which structure is going to reach in the end, had been fixed by DeepMind, by Google, by then Google. And this was a credible feature, but if we see in the training that they have here, there is a layer here in green where they are getting a lot of information from databases. And this information was the only thing that enabled them to get this, on top of their groundbreaking architecture and all the hard work that was in there, of course. But this was the result of decades of getting the information and organizing it and linking it together. And this is where we're going nowadays. We are going for solutions that require more and more data thanks to AI. It needs more and more examples to train to understand what the issue is. And it's not only about that, it's not only about AI. Like, you want to be sure that if you are making a claim, there's data backing it up. This data needs to be accessible to everybody, and a few other terms that we're going to discuss right now. But in any case, the work that people, especially experimentalists, have worked very, very hard to get certain data, the experiments from some, and you name it, is going to benefit future research. And it is very important that we can share it and we can store it. But the way that we do it, it's also important. So there are good practices on how you share the data. So of course, this ensures that the data gets a second life, because if you give data and it's useless, well, good that you store that, but there's nothing that can be done with it. So nowadays, because of this, they're like being able to reuse, to give life a second, give data a second life, has become a requirement for most funding bodies. And remarkably for the European Union, they have made open science and everything that goes with it at the center of all the projects. And what probably is the center of open science is the word fair. And fair stands by findable, accessible, findability, accessibility, interoperability, and reusability. So if we look at more detail into this, we can see, like, we can think on how to make our data more fair, letter by letter. First, findability. You would like to upload your data that you have generated to a known database. That's the first step. It is findable, it is there, but that's not it, of course. But at least it's somewhere. It's not just on someone's laptop that gets lost, broken, and is lost forever. It is somewhere. But of course, once it's there, it needs to be accessible. There's different kinds of databases. The ones I have shown are actually public. Not all of them are. Sometimes for intellectual property or any other reasons, but if you can choose, if your research allows you to have a publicly accessible one, that is greatly increasing the accessibility of your work. Then there's also open access journals, which is free for everybody, for all the students and anyone that doesn't have the money to pay for a subscription to read. And then if you're working with code rather than with actual experimental materials, there is you can upload your code to a Git repository. There's different kinds like GitHub, Bitbucket, etc., where you can put this and make available together with your paper. So you still have the paper. Then interoperability, it means that you can use this data for other purposes. So you can you can maybe merge 100 data sets all together because there's information about it. And the very first thing, the most important thing for this might be the use of standard formats. So at the bottom we see the JSON, and on top of me we see a comma separated values file. These files are formatted in a way that computers can read it very, very quickly, instantly. And that way you can just expand to it. We're going to actually show an example of CSV later. And JSON, for instance, is one of the formats that I use for my tools when I'm creating them. So all the outputs are in JSON and it can be read again. If it's not possible to put it in this format, sometimes data is unique and there's no way of formatting it that way. You still have to make it as computer readable as possible, but you need to provide some explanation about how to read it, or at least that is a good practice towards interoperability. And then for reusability, you need to include all necessary information and need to use this data set again. Sometimes you just see later on we have, for instance, absorbance in an example. Absorbance for what? What was this used for? What was certain information must be there, just to contextualize a little bit more. And well, in this, maybe for an example that's closer to my field, if you're running a molecular dynamics simulation, some of the data that you should put there was which force field did you use for to generate these trajectories or any results that are derived from there, in case people want to reproduce it or just for any other purpose, which might be like merging together, etc. And with this, well, you have fair. And you have we talked about uploading it to databases, and databases are this powerful nowadays because of something called metadata, which is data about data. And this might be about if you have a protein, for instance, if to which organism it belongs, to which family, coded by a gene in which chromosomal region, etc. And that is very important information just to enrich it, but the one that most likely helps the most to interconnect databases and is the unique identifier. The unique identifier ensures that you can reference in your paper a sequence, an entry to a database that you have generated. It can also mean that you can talk about it. Let's say doesn't have to be in your paper, but other people can know that this precise identifier refers to this precise protein. There is no way of making a mistake there. When you talk about that, you talk about that. And then this is used to connect them. We're going to see some example later about how you connect databases with UniProt. We will see that. And then you can enrich, thanks to this interconnection, you can enrich the information of a database with information from others that they are pointing towards each other. This way, well, first of all, findability is very important. Thanks to the unique identifier, you are able to just say, this is my entry, go find it, and it's gonna happen. And also, accessibility itself is usually improved simply by using these databases, because most of the relevant ones are public in any case. So then how the way that you submit to the databases should be as computer readable as possible. Very often they come with their own format. So for instance, it could be based on JSON, which looks like this. You just have the name and sequence and identifiers and so, but sometimes they just have their own one because they used to parse it into the right places. And I also know that in some cases, if you're struggling with where to put the data, certain things you don't know where they go or how to label them, they have a service that they like for free that you can just tell them, well, I put this in this miscellaneous extra thing, it means this, and then they have experts that help you put it in place, or maybe they do it even themselves. Then the use of these formats is very interesting, because the same way that you put it in, it also can be fetched. And that means that for all computational scientists, or even people that just need a standardized way of reading it, it can be fetched again, and then they can reuse it instead of having something that you didn't create and you're not really sure how it's organized. It's all very clearly done that way. So because of that, you can combine data from other sources to your data set, or even you don't generate your own data, you're just putting many things together. And finally, the most important probably, but I don't want to prioritize which one is more important, you can reuse it again. It is it is possible to do that. And with this, we're going to move now to a demo of UniProt that I have recorded, and we can see the different features that it has in there. So there's an example here. When we click on it, we can see, well, to begin with, we don't see the sequence yet. We see a lot of information about it. We see it belongs to human. We see its unique identifier that can be used to be linked to any other database, or just to make reference to it in your paper. And then you have also which kind of protein it is, if it comes from SwissProt, which means that it's manually curated, the length, and well, actually, like if they are certain that the protein exists, which is always the case with SwissProt. So to begin with, you have this function, basically a description about the protein that shows different statements, and it has the sources. In this case, for instance, for whatever statement is here, there's 3 sources that you can just go straight into the primary source, and well, has the evidence for whatever claim they are making. Then you have features that they might have, like domains, binding sites, etc., etc., on top of many other much information from for instance, gene ontology and other databases that might be connected to this, interconnected. So even subcellular location, etc. So things that are interesting about this is that you can even go to, let me just continue down here. If you're interested in diseases, you have here an indication of which mutations most likely cause a disease, and it also shows you where this information comes from which database it comes from. Then what else do we have around here? Post-relational modifications or splicing or whatever might be there. Let's see, because I actually want to show you, like, because it's a bit visual, that you can see also the different X-ray structures that it has. Well, not necessarily X-ray structural files that it has, and you have here so many of them. For instance, I think this is a complex because it's amyloid beta. Yes. So this is how many of them in an experiment that they made for aggregation, I am guessing by the looks of it. They just took the X-ray structure, and here you can see how amyloid beta is aggregating together, and of course you can do stuff with it, which is not the point of this webinar. But what I wanted to show is like all this amount of information, everything you can do with it. You have here the sequence also, you have different variations of the sequence, also depending on where it's coming from. All this is done, first of all, because people are submitting their information. That's the very first step. That's already the beginning that enables us to just even have these databases. But also there's like such intense work from a lot of curators and maintainers in general of the databases to ensure that this stays here, that there's no redundancies, that everything is interconnected between databases. In these cases, they even look for the publications where certain things might be claimed, or at the end there's just so much information in here that when you look at one protein, you might already feel like overwhelmed about it, but then it can get even more overwhelming, because if you just go to the human, you have here all the entries that they have for human, and you go for the reviewed, and then you can see there's like more than 20,000, of course, like all our proteins pretty much, entries here for which you have, I would say, a similar level of annotation. Let's just get a random one and know what this is. You have here some features, and again, absolutely everything subcellular location. What else? Oh, we have here again the variants in case it makes a disease or not. Very valuable information that can be used in very different ways. Oh, look, very interestingly, we have here the 3D structure generated by AlphaFold, the DeepMind method to predict structure. So you have all this very interesting information thanks to, first of all, the researchers submitting it, and again, the maintenance and the structure that it follows. And this database has enabled, for instance, for DeepMind to train the method that was able to do this. So yeah, that's, I think, what's important to have these things, but I'll continue talking about it now. So now we come back from the demonstration, there's some things that you might have seen. The inclusion of all this information that you've seen from all the different databases. Again, it's only possible because machines can read it, and also because the metadata of UniProt is pointing towards unique identifiers of other databases, or maybe the other way around. It also provides something that I haven't shown in the demo for advanced searches. That is not just I need a human proteome, or I need this specific protein. Maybe you need something that is more, I don't know, human proteins that are shorter than this and belong to the family something. You can do all these advanced queries with Sparkle, and it looks like this. In this case, I think it's fairly standard. Like, it looks for, it has to be protein, organism, well, it looks for the taxon. Well, in any case, you can just look for almost any combination possible. And this is very important for crafting data sets and being able to reuse them again. So then now I want to show, because maybe some people are watching this demo, so this webinar, are experimentalists. I have some data that very kindly Roswitha Dolcemaspolo provided, and we used it a couple of years ago for a course on how to make better practices for generating data and a bit of programming. So I have recorded also a little video that I'm going to display here, showing how to get this data from a more experimental friendly view to something that can be used, more reusable, more fair in the future. So we have here, thanks to Roswitha, a data set that she provided with some absorbance experiment. Like, it doesn't really matter at this point with this, but what does matter is that this, if you wanted to put this as an evidence for your article, you just provide this file that has also plots in it, and here a copy of this other table for some reason. This is completely unreadable for us. This is completely unreadable if you want to make some big scale analysis that might be needed for other applications. And also there's not a lot of information. Yes, I see here some concentrations, and maybe with the paper you would be able to find some stuff, but it is not ideal, to say the least. So what I see here is like we have time in hours, we have four different conditions with different substances that affect differently the experiment depending on their design. And also I see 3 replicates, so the time series with 3 replicates and 4 conditions, and it has a blank. So if we wanted to share this, a more suitable way of doing it would be something like this. So with this, what we have is different columns in which it shows like the type of sample it is, if it's like a blank or measurement. I created like different conditions 0 to 3, but that's actually not needed because we have it is explained by these 2 columns, where you have the concentration of I think tetracycline and IPTG, whether or not it was considered an outlier. I think in this case all of them were considered, yes. The time in hours that came out at which the measurement was performed, and also just the absorbance. And of course this is not as nicely readable for us, for humans, as this works better. However, it comes with advantages. And the advantages is, if we go here to the, like I made some code about this data set because we used it for a previous course. If we want to analyze it in a more programmatic way, we can very easily import the data set, and we can already here select the ones that only have measurements and that are only measurements, so it's not the blank. Then we create a new data frame with it. We can very quickly, just with some commands, normalize everything, and we have the blank here. We create like a normalized absorbance, which is basically with the blank removed, and that way from that point on we incorporate, basically we operate with it, and we can go all the way to make plots with it, with the data that's in there. And this can be done at much larger scale with a programmatic way, making a for loop, because that's also why I include the condition. We can do it, if in this case we only have 4 conditions. If we had 100, this would be the same way of doing it, and we could store the plots anywhere. And you can do extra things, I guess, for instance, just like some plotting, there's some warnings, don't worry about it. And my point is, in any case, that you can just play, like you can use this for the same thing that you as an experimentalist would use in Excel, but it also allows some computational analysis after your publication to be done. And just by doing that, one doesn't have to go here and start copying columns and adding some conditions. If you just already provide it in a way that is fair, I guess this is fair, like more fair than the previous one, it's much easier for the data to be reused and to be interoperable with other data sets. And this, in this case, I've made just a tab for Excel for simplicity, just to show you, but this could be safe in a CSV file, which we have here. And well, it looks the same because Jupyter Notebook is just making, Jupyter Lab is just showing it with this. But actually, let me see. Yeah, no. Basically, between each of the values here, you would have a comma that separates the value, and that it's saved automatically. You don't have to worry about it, but it does make life much easier for sharing the data and to reuse it in the future with other maybe data sources, or simply just to check it for just to have it included as supplementary information. This allows the reviewers and other people to just make extra with it. Yes, so with that we are back, and I think by now you probably are convinced, I hope you're convinced, that about the importance of keeping your data tidy in certain formats, and that is, I hope that's true, because then I did my job well. And I want to move into semantic web, because some of the features that we have been seeing from UniProt and we have seen in UniProt, but it's for the databases, comes from this. I am, full disclosure, not an expert on this, and there's full PhD thesis made about semantic web, but I think I can already provide the first insights about how it is. And we go back to the metadata, we'll go back to the example, and you see here that there's a field that is identifier in another database. This other database, well, we start with our own database, and this other database might be this database 2 that is pointed into each other. So we can see here how our database that we are creating is database 1, and that one has a pointer towards database 2, and at the same time database 2 is coming towards to get some information from ours. We also look into database 4, but we're not that interested into it, and there's like this interconnections that at the end, thanks to semantic web, I might be even reaching information from database 3 without knowing about it, but simply because database 2 is connected with it. So yes, that's it. So these connections are done via semantic web, and basically it's done by data pointing at data. So instead of having a website that contains the information, it just contains pointers towards where the information is, and it's fetched when it's needed. And yes, this is done by the use of metadata and unique identifiers, but you might be thinking, which metadata do I need to provide when I generate something? And the question, so the answer, I think, is it depends of which kind of data it is. So are you using a measuring instrument? Put there which instrument it is, and also maybe it's a temperature dependent thing. That's something that should be there, because maybe I'm interested in experiments that only happen above 28 degrees or below. This kind of considerations, you just try to think a bit more expansive as what you need right now, because maybe other people need information that you think it's not relevant. So try to be more in the I gave too much data rather than not enough. Then, well, again, back to proteins, because do that, a reference to the database that contains it, that could be very interesting. So that for sure, you give the UniProt identifier, and for sure you're gonna find it there. And then, but the real thing I would say is like you should be checking before you generate it, because maybe you didn't write down the temperature if you knew that this is interesting. So go spend maybe half an hour in the database that contains the information that you will be generating, and look at what is relevant for that. So that way you can measure and you can be sure that you have everything that is needed. So I wanted to show an example of a Zenodo, like a poster that I created, and I posted it to Zenodo, which is a repository that the European Union has for papers, manuscripts, posters, data sets, quite some stuff. So what I did here is I uploaded the poster. This has a description, has a name, and what is very interesting about this is it shows the, well, first of all, the poster is archived. It shows the authors that you see right under the title, and it has a pointer to their ORS ID, so you can have there everything that they have published. It says the license, I have made it completely open, and where it was published, where it was presented in this case. There is like the meeting, which grant it belongs to, and there you can see RNAct. There we are. Some keywords, so you can look for things that are related. And if I'm making different versions in the future, it also shows up there. You might see it in the bottom right. This I think is very interesting, because maybe you saw a poster in the past and that was the last time that you saw it and had some information. Now you can go and check my poster. Please check my poster. No. So then with this metadata talk, I maybe want to have some closing thoughts about it. Nowadays, science is more than ever about collaboration. And it's not only about, I know this lab that can do this experiment, I go, I learn, and I do my stuff, and I come back and I have a joint paper. You're collaborating with people that you don't know. You're collaborating with people via data sets that they might have generated, or just a single data entry that someone else turned into a data set. And we are in the middle of a revolution, because, for instance, we're not expecting to have the protein structure being solved for almost any protein just like that. And it has happened thanks to artificial intelligence, and this has been enabled by the use of databases. So now it is really the moment to think about, am I doing all the practices correctly? Am I putting everything that I can do to enable the next generation of scientists to do their work? This is now the moment really to think about what fair means and go beyond what the organizations might ask, but really try to have the best practices at the core of your research. Think about what you want to do for your experiments, and think before starting with the experiment, which kind of data is this going to generate, and how am I going to store it, how am I going to share it. If you do that, you're going to be a few steps ahead, because this for sure will become compulsory in the future. So let's start now and make everybody's life easier, and let's advance in science together. Thank you very much for attending to my webinar, and I would like to give some acknowledgements to my group, Bio2Byte on the left, and to RNAct, my university and department, and of course to the European Union for funding RNAct and my project. Thank you very much, and goodbye.
RNAct. To learn more about this project and its progress, you can follow us on several social media channels, with all the relevant information available at www.rnact.eu.